Good morning, children, and welcome to the online teaching session. Students, in our previous classes, we have already discussed about the definition of biotechnology and we have learned some introductory points about the biotech. Once again, we will see the definition. We have read that biotechnology is a process of using living organisms or their molecules or their enzymes just to develop one product or to develop product or to develop processes for the well-being of humankind. So this is the definition of biotech. And father of biotechnology is named as the Paul Berg. Okay, so we can write down the father of biotechnology is Paul Berg. Okay. Now, after this, I have already taught you that why it is important, in which field of the science it is important. Okay. Now we will see what are the steps in case of biotechnology. And I think so the first point I have given you, that is, in the process of the biotechnology, we need enzymes because it is a process to develop something for the well-being of the humankind. If we are going to develop some process, it means we need something, some extra things will be required, and number one thing is enzyme. We need enzyme to conduct the process of the biotechnology. After this enzyme, we need host. Okay. Now, our host should be competent one. Okay. Then we need vector. Okay. I'll tell you the meaning of the vector and everything. And then we need gene. This will also be called as the gene of insert. And at last, we need technique. Okay. So now children, see. In the process of biotechnology, as I told you that it is a process of using living organism or their molecules or their enzyme to develop the processes, useful products. Okay. So just to develop the useful products, what do we need? We need some enzymes. Clear? Definitely in the step of biotechnology, we are going to cut the DNA. We'll do something with the DNA that we will see in detail. So just to use that DNA, just to take out that DNA, just to take out that gene, we need some enzymes. So this is point number one. Okay. And we will develop that enzymes, we will develop that gene into some another's body. And that body will be called as the host body. Okay. So we need a specific competent host so that we can develop our process. Then we need a vector. Okay. Now suppose children, this is your one enzyme, this is your one gene, and this is your host. Okay. And I want that this gene should be here. How to put this? Okay. Suppose this is any of the host. I cannot take this gene in my hand and I cannot put this here because we will see in detail, then we will see what was the reasons that why I cannot put this gene into this body. Okay, because we cannot see the genes, children. So that this detail we will see. But you just think that we cannot put this gene directly into the host. For this we need one medium. Okay, and the medium is called as the vector. Clear? So for this we need vector. Even we can understand, children, by one very common example. We all have seen about the malaria caused by the plasmodium found inside the body of the mosquito. Okay. So how this plasmodium enters into our body? It enters with the help of mosquito. Directly plasmodium cannot enter in our body. It needs one vector, and that vector will be the mosquito. In the same way, if the genes wanted to go to the host cell, then they need the vector, and this will be our vector. After this, we will see what kind of gene it is. If this is the host, this is the vector, then what kind of gene it is. Okay, whether it is useful or not. And finally, what are the processes we are going to adopt? Everything will come under the technique. Clear? So, these are some important points that we have to discuss in the field of biotechnology. Now we will see, we'll begin from the enzyme. See, what is there in case of enzymes? There are enzymes of biotechnology. Okay, it's better to erase this also so that I can give you. So, enzymes of biotech. Okay, number one enzyme is lysing enzyme. Now see children, first thing, we have to isolate the gene of insert. Suppose I have already taught you the application, some points in the application. That is, suppose we need insulin, gene of insulin. Okay, what we will do, we'll take out that gene and we will insert that gene into the host body. This I have taught you. Now children, what will happen? Just to take out that gene, we want to isolate that DNA. And to isolate that DNA, first we will go to the cell. Okay, we will dissolve the cell membrane. Then we will remove the nuclear membrane. We will go inside, we'll take the DNA and then we will cut the DNA into pieces and then we will insert the required DNA into the host body. So these are the steps to take out the gene of interest. So what is there? Lysing enzyme. And the meaning of lysing enzyme is just to isolate the gene of interest. Now gene of interest means the gene that we need to transfer into the host body. So to isolate the gene of interest, this is our first work that will be done with the help of lysing enzyme. And to do this, we'll break or dissolve the different membranes. Now children, suppose if there is a plant cell, then what things are there in the plant cell? We have seen that plant cell is made up of cell wall, then it also contains cell membrane, and cell wall is made up of cellulose and pectin. Cell membrane is made up of, this we have already read, okay, it is made up of two things. One is protein and next is lipid. So what enzymes we need? We need cellulase to break down the cellulose. We need pectinase to dissolve the pectin. Then we need protease. We need lipase. So all these enzymes are required by the plant cell. Okay, if we have to take out the gene of interest from the plant cell, then we need these things to use. Then we come to the animal cell, children. Okay, now I'm going to erase this. Now come to the animal cell. Clear? If I write animal cell, then we all know that animal cell does not contain the cell wall. They have cell membrane only. And the cell membrane is made up of two things, proteins and the lipids. And here we have protease and the lipase for the animal cell. Then suppose if we see the fungal cell, okay, once again the cell wall of the fungal cell is made up of chitin, and to dissolve that we need chitinase. After this, cell membrane, and just to dissolve cell membrane we need protease and lipase. Okay, one more. Suppose bacterial cell, that is prokaryotic cell, and we all know that it is made up of peptidoglycan, and to dissolve this we need lysozyme. Okay, so these are the enzymes, children, that is used to isolate our gene of interest, used to isolate the DNA. Okay, now see, we have one more important enzyme, and that enzyme is called as endonuclease. Okay, so our next point is endonuclease. Now see, we have one more enzyme named as endonuclease. This is the second important enzyme. This enzyme is also called as molecular scissor. Clear? Why it is called as the molecular scissor? Because suppose if this is our DNA, then this molecular scissor goes and make a cut into the DNA and takes the piece of the DNA. Okay, that's why it is also called as the molecular scissor. Scissor means you know very well. So this is the endonuclease. Sometime, or this endonuclease is also called as the restriction enzyme. Okay, so what is the other name? This is also called as restriction enzyme. Now, reason why this is called as a restriction enzyme, because it cuts the DNA at a specific place. It does not cut the DNA at anywhere. It cuts our DNA at a specific place only. Okay, that's why it is called as the restriction enzyme. Now we will understand the mechanism of the endonuclease, that how does this endonuclease work. Okay, now see, our endonuclease is a two-complex system. That is our risk. I am erasing all these things so that I can get the space. Okay, now points are again same: molecular scissor or restriction enzyme. Clear? So it has a two-complex system. Clear? Now the two complex systems are: number one, methylating agent, that is methylase, and endonuclease. Okay, so this endonuclease molecular scissor or restriction enzyme is made up of two complex systems. One is the methylating agent and another is the endonuclease itself. Now suppose, this can be understood better by making one example. Suppose if I draw a bacterial cell here, okay, and one virus enters into the body of the bacteria. Now suppose this is the virus. Okay, and this is the viral DNA. Okay, this is our capsid made up of the protein Viral DNA, we have already discussed these points. This is the plate. Okay, these are the cell endings and all. Now what happens, children? And this is the DNA of the host cell, that is our E. coli. Okay, it is our bacteria. Now what happens, children? This bacteria has its own endonuclease. Okay, now this has its own endonuclease. And definitely if it is an endonuclease, it will made up of the two system. One is methylating agent, also called as FMET, or it's better to write out methylating agent, and another will be its endonuclease. Clear? Now, function of the endonuclease is to cut the DNA. Clear? Suppose this virus will enter into the body. Now, how the virus will enter? This we have already studied by the process of insertion, injection. It will inject its viral DNA and the DNA will enter into the host cell. As soon as the DNA will enter into the host cell, this endonuclease will go and cut the DNA. Okay, at a specific site, suppose AT, at a specific site of A and T. Now, A and T will be found out here also because it is the host DNA, and A and T you can find out here also because this is the virus DNA. Now, in this case, the endonuclease will not be able to identify the A and T position in case of host DNA as well as the viral DNA. So what it will do, wherever it will find that there is the AT, it will go and cut that DNA. But that will be the suicidal case for this bacteria because unnecessary this endonuclease is going to cut at this place. And suppose here is also AT, endonuclease will go and cut at this place. Here also AT, endonuclease will go and cut. So this will be the suicidal case for the bacteria. And this endonuclease will also cut this. But it is of no use now because the bacteria has already died. So what happens, children? As soon as the endonuclease will go and cut at these sites, there comes one more substance, one more complex, and that is methylating agent. Now, methylating agent is a self-defense mechanism. Self-defense mechanism for this bacteria. So, what this methylating agent will do? This will go and bind at this site. Okay, this will go and bind at this site. Go and bind at this site. At one of this site, A and T. Clear? So, as soon as there is a cut, methylating agent will go and it can either bind to A or T, and it will prevent the bacterial DNA because endonuclease can identify A and T. But now there is no A and T, as wherever there was the places of A and T, methylating agent has come and bind it either side. So now endonuclease is not able to identify the gap between the A and T. So it will not cut the host DNA. What it will do? It will directly go to the viral DNA and it will make a cut over here. Okay. So this is the self mechanism of our E. coli bacteria to prevent itself, to prevent its DNA from the host DNA. Understood? So this is all about the endonuclease. Clear? The rest portion of the endonuclease, that where it cuts, how it cuts, that we will see afterwards. Clear? So till here, children, you prepare this. Okay. Now, yes, till here you prepare this. Rest we will see in the next class. Thank you.